Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base biotech podcast, and this is number 55, and it's going live on April the 17th. Because it's getting toward the end of April, any day now I'm expecting the Halloween merchandise to hit the stores. Or maybe they'll just leave the Easter stuff out for Easter 2027. Last week we had a conversation with Dieter Wilbold, CEO of Preavoid, and Antje Vulleveit, Director Preclinical Development, also at Preavoid, about Parkinson's disease. And we chatted with Karsten Eastman, CEO and co-founder of Cythera Therapeutics, about a new way to build more stable and drug-like peptides. This week we're talking to Jason Bock, CEO and co-founder of CTMC, and with Eledon President and CEO Steve Perrin. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us, sponsoring a podcast episode, or for event coverage. First, it's time to go through some of the headlines of the articles you may have missed over at deeptechdigest.com. Select Precision's nanomedicine is set to target the most challenging tumours. Fundraising is taking place for Lennox-Gastaut syndrome research, and a T-cell therapy enhanced with membrane-tethered IL-12 booster has received fast-track designation. Gilead is acquiring Tubulis. There has been validation of an AI platform for an opioid use disorder treatment, and a new company has exited stealth to advance sonogenetics into the clinic. We had an article on supporting biotech innovation in Singapore. A new formulation of an SCD1 inhibitor can reach the brain, and breakthrough data show complete tumour eradication and durable protection in an aggressive brain cancer model. There was an article on delivering healthy mitochondria to diseased cells. A new study shows the increasing value of real-world data in clinical trials. And Fortitude Biomedicines announced its axial spondyloarthritis lead program. And you can read all of these and plenty more at deeptechdigest.com. Right, let's kick things off with a company that declares it is much more than a CDMO. CTMC is a joint venture between Resilience and the MD Anderson Cancer Center, and it was created to accelerate the development and manufacturing of impactful cell therapies for patients with cancer. To tell us about CTMC is its co-founder and CEO, Jason Bock. Hi Jason, pleasure to have you on the podcast today, and I wonder if to get things started you could first tell us a bit about CTMC.
I could tell you, Jim, maybe with the origin story of a more than 25-year career in small, medium, and large-sized biotech and biopharma, developing novel products, bringing them from research into the clinic, and then fortunate to be involved in bringing several products through to global commercialization with peak market sales of over $3.5 billion. That was a large pharmaceutical company. And so about 6 years ago, saw all of the exciting results in oncology, in cell therapy, CAR Ts, and yet saw the challenge in how those were going to be developed in terms of both manufacturing challenges and just how that whole supply chain was so different than we were used to for more traditional modalities, even complex, what we considered complex modalities back then, like monoclonal antibodies, which I spent most of my career on. And so at the same time, some guys from leadership from MD Anderson reached out and started talking to me through friends of friends And so we came up with this idea of trying to create a different kind of organization that was specifically designed to meet some of the challenges that we could see emerging in cell therapy. That's when I moved to Houston, took a role within MD Anderson in a division called Therapeutics Discovery, which is like an internal biotech within MD Anderson. So built with industry experienced people. No one has academic titles, no tenure. And with the idea of partnering with academic researchers to industrialize their products, develop them in a biotech type style, could run clinical trials at MD Anderson, one of the largest clinical trial infrastructures in the world, and understand whether we could get products to clinical proof of concept, and then decide where to go with them at that point. Commercialization deals with larger companies, spin out to biotech and the like. And so that's what we started on doing as employee number one in that group. And we were very excited to be able to identify the facility I'm sitting in today that was built and owned by a CAR T company called Eledon Pharmaceuticals. They had gotten to a stage where they had built a beautiful facility, and yet it was a little too big, honestly, for the pace of their product development and their portfolio. So we made the interesting maneuver for MD Anderson to acquire this biotech facility. It's about 60,000 square feet, 14 GMP clean rooms, quite new and quite industrially built, quite large clean rooms. And so that's when really CTMC sort of got founded, when we actually had space and people. And what was interesting, Jim, is then we created an engine not only for those academic researchers to have a way to industrialize their products different than an academic GMP, very much like biotech grade, but a number of biotech companies, venture capitalists, CEOs, board members reached out to us who were sitting in biotech companies, cell therapy, early stage cell therapy companies, were like, hey, what are you guys doing? Could we find some way of working together? And so we developed a whole development model, co-development model, where we utilize our infrastructure in terms of process and analytical development, regulatory, we had a full regulatory group, and the clinical infrastructure within MD Anderson to offer really, a lot of people say this one-stop shop, but this is really one-stop shop where we could partner with companies that had an interesting product that was in research or ready for IND-enabling studies, and essentially do everything required to carry that product through to 15, 20, 25 patients or more of clinical data. And so that model became very popular, and we signed a number of partnerships with biotech companies to help them develop their products. And in fact, over the last 3 years, together with our partners, we've developed and filed INDs on 9 different products. So it's been quite proficient, and we've been able to do that with speed. Overall, a very capital efficient model, and with relatively low risk, because we're doing things at very high standards in manufacturing, and then we're running clinical studies at one of the premier institutes in the world in terms of developing novel products and seeing how well they work in patients in terms of MD Anderson.
And I noticed on your website, it makes the bold statement that you're more than a CDMO. I wonder if you could explain that.
Sure. That's partly why I started with the origin story. We didn't set out to say, hey, let's build something that could manufacture things under a contract for people. It was, what's the problem statement? Problem statement is there's a different kind of supply chain required for running autologous cell therapy trials. They're really complex. There's an evolving regulatory landscape that's challenging to operate within. And clinical trials are very difficult to execute because the source of the starting material comes from the patient. And then you have to manufacture in real time under huge time pressures, get the product back to that same patient. And so those are very different challenges than we've seen for other therapeutic modalities. And we specifically built the skill set and the capabilities at CTMC to specifically address those challenges. We develop regulatory strategies, execute those for our partners. We, in fact, can write the entire pre-IND and the IND. And then in connection with MD Anderson, we can execute the clinical trial with minimal support from the biotech company. And so a lot of the companies we work with can stay relatively virtual, 5 people, 8 people, and they have a clinical stage asset and are executing a world-class clinical study at MD Anderson because of the model we've set up. You can't do that if you're just a CDMO. And it's not disparaging at all for some programs. A CDMO model may be just right, but for many companies, being able to stay smaller, not create a large internal infrastructure, not have to go out and find these very specific talent sets, we can offer a lot of opportunities for them.
You were mentioning earlier about how you've been able to do quite a few things quickly. How do you accelerate or help accelerate that path to commercialization?
Everything we do is designed to get to value inflection milestones quicker with less risk. And so, for example, a lot of organizations use the milestone of time to IND, right? And that's very important. And so we're very good at that as well. Understanding when to get regulatory input, what questions to ask. We actually have more, I think, experience than any other group with early stage cell therapies because we've been able to file all these INDs and work with our partners on that and that learning. So first is you can lose a lot of time in regulatory interactions. And so we've been able to really streamline those interactions. But then you get to your IND, FDA, you get your safe to proceed, and in 30 days, you want to start dosing patients or thereabouts. But then there's this whole infrastructure need to set up and execute clinical trials. That's even more complicated for cell therapies when you're bringing in the product and where you're going to store it and how are you going to thaw the product before administration and all these things. And so typically, it can be at least 6 months in between IND filing and what's called clinical site activation. Some small specialized sites can do it a bit quicker. MD Anderson, large clinical sites sometimes are actually a little bit slower to set up the initial trial, but then the enrollment goes very quickly. But because of our relationship with MD Anderson and the length of time we've worked with them, the number of products we've put through that system, we've now streamlined that whole exercise. And so we can get from IND filing to site activation in our target is less than 100 days. We've always been able to hit our target, sometimes downwards of 70 or even 65 days. And so lots of people talk about speed. And I always say that we don't actually pipette faster than anybody else. Cells in our incubators don't grow any faster than anywhere else. But we've engineered some systematic elements into our development model to streamline the development and enable shorter timelines. That time from IND to site activation, there's a lot of paperwork and a lot of committees, meetings, and things like that, but there's really no value being created for the therapy. And so we've been able to focus on streamlining that, starting that clinical trial sooner, and getting to clinical proof of concept, which is really the value inflection milestone that we're targeting and that really helps biotech companies. We want to see how well the product works in patients as soon as we can. Do everything in our model to enable that.
Clearly, partnerships are at the core of the company. I wonder if you could tell me about the partnership with Link.
Okay, sure. Partnerships are what we're all about. A large part of what we do is partner selection. It's definitely a two-way street where, of course, companies have to want to work with us and buy into the model we've developed. We have to be interested in working with them, which includes finding a clinical PI who is not only like, oh yeah, that seems like an interesting concept, but we're going to put a lot of pressure on them to enroll patients and find the right patients And we want the clinical PI who's like, I am super excited about that trial, and I will be an advocate and I will find the best patients and put them on the trial. So they're actually pulling the product from us because we start talking to them before we've even signed a partnership. And so during the IND enabling studies and things, the PI is an important part of the partnership of giving input into these are the kind of patients I see. Even, Jim, which is really important, providing us relevant patient material to do the development work on. Is a little bit outside your question, but I think it's an important point is that often goes missing with cell therapies. As we talked about earlier, the starting material comes from the patient, but patients are not homogeneous, and especially cancer patients that can be, depends on their prior lines of treatment, depends on the indication. And so if you have a patient sample from melanoma patient and you're going to be running a study in renal cell carcinoma, it may not be that relevant. But even worse than that, most of the development work that's done in cell therapy is on healthy volunteer material because that's what's available. We have a great inside track in working with the MD Anderson clinicians and getting relevant patient material to do our development work on. So there's less surprises on manufacturing when we actually get into the clinic. So that's a little bit on like what we have structured things to be good partners to biotech companies. And then biotech companies, we're looking for breakthrough novel concepts. So a lot of what we're doing now is working with cell therapies in trying to address solid tumors, which represent 90% of all cancers. So that's where is the greatest challenge, but we think we can have the greatest impact. And then the business side is that our partnership model is very much, we're all in this together. And so we structure it in a way that has us take a fair amount of risk. We offer our partners basically a fixed cost of development from where they're starting to a milestone. Typically, that milestone is like 10 patients dosed in the clinic. And so we're taking a product that's at a research stage and doing everything needed to get to 10 patients of clinical data, 15 patients of clinical data for one fixed cost. It's typically in quarterly payments and is all in, includes raw materials, includes shipping costs, includes everything. So what's great about that for biotech companies and what creates powerful partnerships is that we're in it with them. Our incentives are completely aligned to get to those milestones as quickly, efficiently, and robustly as we can. If we need to do another experiment to make sure that we've really got the process down, then we do that other experiment. And the biotech company, their costs remain fixed throughout that period. So the CFOs really love the model because they have complete costing visibility for one of their biggest costs they have in drug development. The other thing that enables, really in terms of partnerships, is that because we don't have to go back when the technical team identifies a particular change that needs to occur, we need another set of experiments, we need to look at a different variable or something. You don't have to go back to the business guys and like, okay, let's price this, let's come up with a change order, let's decide how much it's going to cost, let's send it to their business guys, let's negotiate. During that time, usually you're losing time. Because we cut out all of that stuff, the technical teams just work really hand in hand to rapidly develop the best process, weighing all the challenges that we all have to weigh in drug development all the time: speed, quality, risk, cost, and things. And so balancing all of those, but we're completely aligned in trying to assess those. And I think that's another secret of how we've been able to accelerate timelines, mostly because lots of times timelines get slowed down by all these other negotiations and stuff, and we've eliminated all of that.
Because of those partnerships and companies that you work with all being so different, what are the biggest challenges associated with that and trying to do all of this efficiently and quickly?
Yeah, autologous cell therapy is hard in general. I love it. I love doing hard things. Biggest challenge for us is we're supporting now a number of different clinical trials. All of them have different enrollment rates, and they're all somewhat unpredictable. because it's when the doctors find a relevant patient, then we open up a manufacturing slot and produce that batch. And so getting the cadence right to be able to be flexible. One of our corporate values is nimble. So being nimble to be able to adjust to the clinical enrollment of multiple ongoing studies. I think honestly, we have a better opportunity to do that because we have a wide portfolio, and typically when some projects are enrolling quickly, others may be a little bit slower, and we can balance those things and been able to balance those quite well. But I would say that's an inherent challenge in cell therapy development and one that we've been built to address.
And so what's the CTMC Network Alliance?
You know, Jim, as we developed this model of industrial manufacturing connected with a major academic medical center, it like resonated with lots of people when I go and talk with them. And the medical centers are really one of the strongest advocates for cell therapies. These are transformational for my patients. And as our model became more well known, number of academic medical centers from all around the world reached out and said, hey, can you explain more how this CTMC MD Anderson model works? Because everyone's been trying to crack it. And I think it's been really hard. A lot of the approaches have been from academic medical centers who have very sophisticated stem cell transplant groups that then are a natural bridge to cell therapies. It's just that they're typically more focused on very academic type processes, very early research processes, and aren't as familiar with more like late stage development or what do you need to do in early stage to prepare yourself for late stage. All of these things that typically biotech companies are more comfortable with doing, and that's why early research spins out into biotech. And so we definitely hit a chord between this industrial manufacturing and academic medical centers wanting this access to this capability. And the rollout for cell therapies internationally has been slower than anyone wants because these therapies can impact patients' lives, and yet accessing them in a short enough time to be relevant for patients has been a challenge. And so what we came up with was CTMC Network Alliance, which is basically an umbrella of us working with major academic medical centers around the world to help give them access to cell therapies. And that can take a number of different forms. The one that's most advanced is our partnership with Albert Einstein Hospital in Brazil, one of the most innovative hospital networks in the world. And we have a partnership with them to initially work with their existing cell therapy infrastructure that focuses on CAR-Ts and do everything they need to train them up on how to manufacture TILs. TILs are a proven modality for solid tumors, initially in melanoma, but require some very specialized techniques. And as well, that CTMC has developed some proprietary reagents that make that TIL process more robust. Typically, there's a surgical resection that's needed to start a melanoma manufacturing process. You actually have to take out a chunk of tumor from a patient, and then we're able to extract the TILs from that chunk of tumor. With some of the technology we've developed in conjunction with MD Anderson, we're able to start with a core needle biopsy rather than a surgical resection. And so that whole package of know-how and reagents and training, we partnered with Einstein on in order to provide them the capabilities of running TIL manufacturing locally for them. And our goal with that is to enroll and treat the first patient with TIL in South America. So a pretty ambitious goal. And then we have conversations, pretty advanced conversations ongoing with many other academic medical centers throughout the world to help meet them where they are and essentially increase access to advanced technology required to deliver cell therapies.
How would you say automation is affecting the industry?
Yeah, I think that's an area where it's coming and it's impacted some unit operations so far. But I think it's an area which is imperative that we accelerate. This is for later stage cell therapies and for commercial, where because they're personalized treatments and we have to make one batch per patient, there's just a lot of batches. And with manual processes, that requires a lot of people. So we have to scale out, not scale up. And I think the hardest thing to scale out with is people. The necessity of training thousands of people to make thousands or tens of thousands of batches is just a real challenge for the industry. And so I think the interesting hope is that there's a convergence that will occur with the advent of physical AI and the rapid uptake of that, where we can substitute some of the labor with automation through robotics. And so that's a whole nother topic, but something I'm very interested in, that I think there's this huge and growing need in cell therapy. And from a different angle, there is a massive progress on physical AI. That's the next big wave. And so structures and organizations that can capture that convergence will make a huge impact on the scalability and access to these treatments. And as well the cost, but I think mostly the scalability. If we had all the money in the world and we wanted to make 100,000 cell therapy doses a day, we couldn't do it. And most of the reason we can't do it is there's not enough people. Either we can start training tens, hundreds of thousands of people, or we can automate and roboticize processes. So I think that's going to be a huge driver going forward for cell therapy.
And what about the difference between countries? We see lots of news on what China's doing in manufacturing and within the industry. How does that affect what's happening, say, in the US?
Yeah. See whether I can draw this analogy. I haven't done this before, but China has done with cell therapy a little bit like what happened with mobile phones. The way it worked in the US was we had telegraphs, then we had phone lines, landlines, if we remember those, then we moved to mobile. China was able to sort of skip over that whole landline and was a really early adopter in mobile networks and mobile phones and things. And a little bit the same way in that, the US really dominated in small molecules and antibodies. China started to really advance in antibody therapeutics over the last 10 years. But then in cell therapy and gene therapy really had a running start. And so has been neck and neck with the US in terms of innovations in cell therapy and development of those products, over the last 5 years. And I think the ability to integrate manufacturing, clinical, and regulatory is what is enabling China to do things very quickly, and even US companies for tapping into that capability of bringing those three domains together. In fact, in China, for studies run through a hospital, it's actually not a need to even file a version of an IND. You can just get hospital approval to treat patients. And so that has the opportunity for streamlining development. So it seems like over the last year or two, there's been a great awareness of these development capabilities in China. If, Jim, you're able to see and connect the dots, a lot of the things that enable China to move quickly in development were some of the founding elements of CTMC in terms of connecting manufacturing with the clinic within a regulatory framework. And so a lot of the advantages that China has over sort of traditional development in the US, we've already captured. I think, to be fair, though, one aspect that is still very existent in China is cost of development. I think those academic medical centers and companies are quite subsidized by the government, and so they're able to offer quite low costs. I think the challenge is for drug development, especially early stage drug development, is a lack of control that companies have in the execution of those trials, especially American-based companies sort of outsourcing their initial manufacturing and clinical trials to China. One example we can talk about that's very timely is some recent clinical data that was published, actually an exciting clinical study on in vivo CAR T for multiple myeloma that was recently published on the first 5 patients who were enrolled in a clinical study done by a company called ESO. And they have subsequently partnered with AstraZeneca. And so the first 5 patients showed pretty good responses, fairly deep MRD type responses in multiple myeloma with a in vivo anti-BCMA CAR T. However, there were quite a bit of toxicities. All the patients had quite severe CRS. And unfortunately, one patient died on the study about 2 weeks after dosing. And all of them had, from the CRS, quite significant immune responses. And so that trial was stopped after 5 patients. Clear technological milestone. I mean, I think it's pretty amazing that we can take a lentivirus encoding a CAR, infuse it into a patient's bloodstream, and get enough of a response through infected T cells to eliminate a lot of the multiple myeloma, the B cells. I think that's amazing, like just from a technology point of view. But from a drug development point of view, having a study where you have a new modality, you treat 5 patients, you basically have to stop because of toxicities, isn't a great outcome from therapeutics. And in fact, it opens up a lot of challenges. One of the biggest challenges in new modalities is how do you deal with toxicities, especially when you're, essentially infusing a patient with something that is designed to infect specific cells. But what happens when it infects different cells? What happens when you get few percentages of a CAR expressed on endothelial cells or other cells? And what's going to happen is you're not actually going to know that. All you're going to see is some toxicities. And then you're going to have to try and figure out what they are. But when you don't really know what cells you're infecting, I always thought that one of the challenges with in vivo CAR is long-term, we would see some mysterious toxicities and would try and be hunting down, what caused those. Wasn't honestly expecting to see such short-term toxicities. They saw a huge immune response after the infusion, the immediate infusion, the lentivirus. One patient, it was as soon as 2 hours and had a very severe immune response. And then there was a second wave of the immune response presumably triggered by the CAR. And so I think there's potential that this actually really slows down. These results actually slow down instead of speed up in vivo CAR T field, because that product is going to be, I think, difficult to develop further. This is how, Jim, I would tie it together with your question about China. Like, okay, so you dosed these 5 patients in China. You did it quickly for low cost, which is great, got the result. Then where do you go from there? Do you lower the dose and do another study in China? Do you lower the dose and do a study in the US? If you're trying to enroll patients, you're going to have a tough time. The efficacy wasn't as good as ex vivo CAR T. It was good, still impressive for like first 5 patients, but not as good. So who's going to enroll in that study? I think now you're going to have a tough time. And so, with cell therapies, one of the benefits of developing them is you can find out fairly early in clinical development how well your product works. It's amazing that we can get responses in first few patients. We see that with ex vivo CAR T or even other kinds of cell therapies, that in the first 15 patients, you pretty much know if you're how well your product is working. But on the other hand, you pretty much know how well your product's working. And so if you get off track in your first 10 patients, it's pretty hard to get back on track. It's going to be really interesting to see how the field develops, both on the axis of ex vivo versus in vivo, as well as the China-based development.
Is there anything that you wanted to mention in the context of what we were talking about that you would like to talk about?
Sometimes when it's darkest is when you need the most persistence and to just keep moving forward through this. We know it's unequivocal that these therapies are impacting patients in dramatic ways, and that patients are walking around today that wouldn't be without these treatments. And so that is unassailable. And there are a lot of challenges to address. I love challenges and things that are really hard with a fantastic outcome at the end. So at CTMC, the guys here work so incredibly hard and diligently to drive product development forward, to find ways of saving time. And we are making an impact for what I think is a quite noble cause.
Great to hear about the work that CTMC is doing, and a little about China as well. We've had a few kidney interviews recently, but this one goes beyond that. It's not only about kidney transplants, but also the impact of a drug on diabetes and other transplants. Eledon Pharmaceuticals is the only company developing an immunosuppression drug that targets the CD40 ligand pathway. To tell us more is the company's president and CEO, Steve Perrin. Hi Steve, thanks so much for being with us today. Really appreciate it. I wonder if first you could tell us a bit more about the company.
Yeah, sure. Happy to. And thank you for inviting me to the podcast today. So Eledon Pharmaceuticals is a biotech company that has been focusing on the development of a monoclonal antibody called tegoprubart, which targets a specific receptor on immune cells. And we've been focusing the development of tegoprubart in the prevention of transplant rejection, initially focusing on kidney transplant rejection because it's one of the biggest indications both here in the US as well as globally as far as the number of transplants that are done. But conceptually, tegoprubart could work in any type of transplant, any type of cell, any type of organ, and it even doesn't even matter what species it might be. We have shown that we can protect allotransplants, which is human to human, but we also have been in many of the xenotransplant studies, which we can talk about, which tends to focus on the utilization of organs from other species into humans. So tegoprubart, our goal and mission as a company is to replace the standard of care medicine for transplant rejection. It's a calcineurin inhibitor called tacrolimus. It's a 30-year-old drug, and it's quite effective in the first year at preventing transplant rejection, but it has a lot of both short-term, immediate, and long-term toxicities, including cardiovascular toxicity and hypertension. About 30% of patients on tacrolimus will get type 1 diabetes because it kills the islet cells in your pancreas. It has nephrotoxicity, so it's actually toxic to the kidney that it's trying to protect. And it has neurotoxicity. It causes severe Parkinson's-like tremors, which are quite debilitating to patients. So our goal is to replace tacrolimus as the cornerstone medicine to prevent transplant rejection.
And what are some of the challenges that are typically associated with transplants? And I include in that, I guess, a shortage of transplants.
That's probably the biggest issue from 10,000 feet. Is you really articulated quite well what the issue is. There's over 100,000 patients on the transplant waiting list here in the US for a kidney. And that's really driven by the fact that the current standard of care, although it works quite effectively at preventing rejection in the first year, the 10-year organ survival is only 50%. And although that sounds really like a long period of time, it's actually not. Many people on the transplant list are in their 20s or 30s due to chronic kidney disease. And so with organs only lasting 10 years, you can imagine that a 20-year-old may need 3-4 transplants in their lifetime. And in fact, the vast majority of people that are on that waiting list that will never get an organ, period, are there for their second or third transplant. So we're trying to solve for that by really extending the long-term function and survival of transplanted organs. We think on tegoprubart, because of the fact that it doesn't have those toxicities that I mentioned, it should really extend long-term organ function and survival.
And what's the issue with having to have several transplants?
So because of the fact that tacrolimus is toxic to the transplanted kidney, when one loses their kidney over time due to loss of kidney function, you end up having to go back on dialysis, which has a 5-year morbidity that's quite challenging. So dialysis is very, very difficult on people. And the older you get, the harder dialysis is because it ruins your circulatory system and people can't find veins for your dialysis that you need to do several times a week, either on site or at home. And so it becomes quite problematic for patients that are on dialysis for long periods of time with comorbidities. And yet they need to be on the transplant list to get their second or third organ, which could take years to get a match.
Does being on dialysis for a longer period of time and couple that with the wait for the transplant, does that affect how well the transplant takes?
It can because of the fact that you're impacting your cardiovascular system over long periods of time. So when you get your transplanted organ, it may not be as effective, especially if you're on your second or third transplant. The other thing about current standard of care with your transplant, we often measure kidney transplant function by a metric called eGFR. It's basically a measurement of how well the transplanted kidney is filtering. And a perfect eGFR for healthy individuals that are young is around 110 or so. As we get older, we lose a little bit of function and the eGFR can drop into the 90s. But people that are on dialysis literally have no filtration capabilities whatsoever. People that have had a transplant but their kidneys are starting to dysfunction will start to get into the 20s and 30s as far as an eGFR and need to go back onto dialysis. Ironically, because tacrolimus, that drug that's trying to protect the kidney, is toxic to the kidneys, a freshly transplanted kidney on TAC only has an eGFR in the 50s typically. That's about the average after transplant. So you can see that even though it's a kidney that just got transplanted, it's not functioning as well as a normal healthy kidney. The amazing thing that we've seen from our clinical trials using tegoprubart instead of TAC is our eGFRs tend to be in the 70s. So we're at least 20 points higher than current standard of care. And eGFR predicts long-term kidney function and kidney survival. And that's why we think tegoprubart is going to result in kidneys lasting a much longer time than current standard of care.
It's slightly off topic, but how do you improve the number or increase the number of people that become kidney donors? And clearly there's a correlation between donation and the lack of transplant organs.
It's a great question. I mean, certainly education is one of those mechanisms to increase the number of donors, especially I think here you're referring to living donor kidney donation. The vast majority of new kidney donations in the last 10 years has not been driven by living donors. That's been a kind of a flat line. And so the only way to improve that is educational processes to educate people about the safety and the amazing gift that you're giving to be on the donor organ donation list and to be giving a kidney if you can. The increase in kidney transplants over the last 10 years has been driven by a new type of kidney that we used to not transplant a decade ago. Most of the time, kidneys that are transplanted are derived from brain dead donations. As of late, we've been starting to use cardiac death associated kidneys, and that's because what we call perfusion technologies, when somebody dies, they perfuse the body before harvesting the organs in cold solution to make sure the organs are of good quality for transplant. Those technologies have gotten much better. And so we're seeing a dramatic increase in donated organs from cardiac death because of the perfusion technologies. Now, the only real way to shorten the wait time for an organ, if you want to call it that, there's only two ways to basically shorten that average 5-year wait time and decrease the list from hundreds of thousands of patients waiting. There's only two ways to do that. You can either make the organs last longer, and we're trying to solve for that by a better drug like tegoprubart, which will make organs just last longer than tacrolimus. And the second way is xenotransplant, another source of organs. If we don't have enough human organs, can we use organs from genetically modified pigs as an example? And that's a new field that's been developing in the last few years.
Speaking of which, I wonder if you could explain a little bit more the difference between allotransplantation and xenotransplantation. I mean, not a little beyond the definition. What are the characteristics of the kidneys if they are from not a human source?
Sure. So as you mentioned, allotransplantation is within a species. So if you're working in preclinical experiments in the lab. That could be a mouse to a mouse or a monkey to a monkey. And in the case of humans, it's human to human. That's the definition of allotransplant. Xenotransplant is a transplant across species. So it could be putting a mouse organ into a rat, as an example, in preclinical. In humans, at one point in time, many, many years ago, we were trying to put non-human primate organs into humans, but the size matching was a little bit more challenging. So we moved to pigs as the most common source of organs that one might be able to use for xenotransplantation into humans. And that's because we can control the growth of pigs. But more importantly, the holy grail would be to humanize a pig with genetic modifications so that if you have enough humanization of the pig, you could take its organs, put them into humans with less immunosuppression. And that's where the field of xenotransplant has really been focusing in the last 10 years with companies like United Therapeutics and eGenesis. They've been making genetic modifications to the pig genome to try to make the organs more tolerable when we transplant them into humans.
What about the potential for growing organs in the lab or 3D printing of organs? Is that a potential in the future?
It certainly is a potential, and it's been something that the scientific community has been working on for a significant period of time. I think the complexity there is the three-dimensional architecture and the number of cell types that are present in most organs. Kidneys being a very complicated one, the filtration or glomeruli of a kidney is made up of multiple cell types, each of which has to communicate with each other, including hormone-secreting cells that modulates the body. So a kidney is a three-dimensional printed organ, might be quite challenging. Contrast that to other organs, or like the heart. The heart is a pump. It's mostly muscle. So there might be some advantages depending upon organ selection when you think about 3D printing or matrix-based in vitro structuring of an organ. Some might be easier to do than others, but we're probably still a little bit off, except for in the context of wound healing, where we've made some progress there.
So could you maybe tell me a little bit more about your drug and how it works?
Yeah, tegoprubart is a monoclonal antibody, as I mentioned. It targets a cell surface receptor on an immune cell called T cells. It's a key regulator that controls what we call both adaptive and innate immune signaling. So it not only can modulate the activation of T cells and prevent their pro-inflammatory activation, but it also can control another population of cells called B cells, which would make antibodies to a foreign organ. So this pathway that tegoprubart blocks, the receptor is called CD40 ligand. It's on the cell surface of activated T cells. It was identified back in the 80s, and it became clear that modulating and inhibiting this pathway was the most effective way to prevent transplant rejection in multiple species, initially shown in mice and rats, later on in multiple non-human primate species. And today, obviously, Eledon is focusing on the development of tegoprubart in prevention of human transplant rejection.
And you mentioned earlier that it's applicable to different kinds of transplant. Is it equally effective with all of the different transplants, or are there differences?
In theory, the mechanism of action of tegoprubart, it should work across any type of transplanted organ or cell, and it shouldn't matter if it's an allotransplant human to human. It should also be quite effective across species, including xenotransplant. Now, we're collecting a lot of clinical data along those lines already, Jim. As I mentioned, we reported our phase 2 clinical results of our BESTOW study last November at Kidney Week. The data was quite encouraging, where we had better EGFRs than standard of care. More importantly, though, we had an incredibly better safety profile. 30% of the patients on TAC got new onset diabetes. We didn't see any on Tego. There was increase in hospitalizations due to hypertensions and severe cardiac events in the TAC arm. We saw none on Tego. There was 9 times more tremors on the TAC arm than Tego, and our EGFRs and kidney function were better on Tego compared to TAC. So it was incredibly exciting data with some of the best EGFRs that's ever been reported in a clinical study. We were in the mid-70s, which was really exciting since that predicts long-term kidney survival. We also were in the clinic in an investigator-initiated study in University of Chicago with islet cell transplant for people that have really severe type 1 diabetes with what we call hypoglycemic unawareness events, where they can't control their blood glucose swings. And that leads to severe comorbidities for those patients, where they can pass out when driving a car, or they can pass out in their sleep and actually die because they're unaware of it and they're not taking their insulin to control their blood sugar levels. So what we've been doing with Dr. Wachowski at University of Chicago is he takes this patient population with really severe hypoglycemic unawareness events, and he does an islet cell transplant. He isolates islet cells from the pancreas of a deceased donor organ, and he transplants those cells into those patients, and then he protects those cells with tegoprubart. His first 10 patients got off exogenous insulin within 30 days post-transplant. And these have been sick people. They've been on insulin for 30, 40 years, really poor quality of life, have to be very careful about what they eat, how they exercise. And within a month, they were off exogenous insulin and making their own insulin with the transplanted cells. Some of those patients are now 2 years out post-transplant and actually doing just incredible. Now, islet cell transplant's not new. It's been around for decades. But the problem with islet cell transplant historically has been people have had to use tacrolimus to protect the transplanted islets. And as I mentioned, tacrolimus is actually toxic to islet cells. And so when they were doing islet cell transplants using tac to protect the cells didn't have very good metabolic function. They couldn't get people off exogenous insulin as quickly. And Dr. Wachowski has really commented that tegoprubart is going to revolutionize the ability to do islet cell transplant in patients with type 1 diabetes, which is really exciting.
And in transplants such as kidney, when you have people that are maybe pre or going in for their first transplant and third transplant, are there any differences between the results that you're seeing in those patients?
So far we've done about 100 kidney transplants, but they've all been what we call de novo transplants, so people receiving their first transplant. We have not investigated the effect of Tego on second and third transplants yet. We are working with the agency and FDA and EU in the first half of this year to design and plan what we would call a pivotal phase 3 study. This would be a global study in de novo kidney transplant. Again, people getting their first transplant. It's probably going to be about 700 patients. It's probably going to be multiple different geographies, including Europe, South America, North America, including Canada, as well as Australia. So a fairly big clinical study. And it'll have a very similar endpoint design to our phase 2 BESTOW study, which is what we call a composite endpoint of patient survival, graft survival, and the percentage of biopsy-proven rejection, all at 12 months. So we're hopefully going to launch that study this year.
Right. And where are you at on the clinical trial pathway and the different studies that you're doing?
So for the kidney study that I just mentioned, we're in the process of setting up a meeting to discuss that phase 3 clinical design plan with the FDA. Once we do that in the first half of this year, we're hoping to launch that pivotal phase 3 study by the end of the year. In islet cell transplant, as I mentioned, Dr. Wachowski has now enrolled 12 patients in his study. That data is quite encouraging, and Eledon would like to take that study over and start to get FDA approval and alignment on what's called the phase 1, 2, 3 bridging study, using cadaveric-derived islets with tegoprubart for that patient population with type 1 diabetes. We continue to support xenotransplant globally. I mean, we've been working in xenotransplant in non-human primates with collaborators all over the world, several different sites here in the United States, one in Europe, a couple in China. And as you probably have seen, we're starting to transition the use of tegoprubart as one of the cornerstone medicines in xenotransplant and in the human studies. We've done 4 or 5 patients at Mass General Hospital that have received pig kidney xenotransplants. We did a cardiac transplant at University of Maryland. And the agency has now gotten alignment with some of these companies that have made the genetically changed pigs to start phase 1, 2 studies at multiple centers. So I'm sure that we'll see many, many more xenotransplants occurring in the next couple of years.
You were recently at the Advanced Technologies and Treatments for Diabetes conference. Could you tell me a bit about what you presented there? Because as you were saying, it seems like it's going to be massively transformational in that condition.
Yeah, so Dr. Wachowski there presented an update on the patients that I described that he's done to date. The focus there was an update on the first 10 patients that I had mentioned got off exogenous insulin within 30 days post-transplant, how well that they were doing. None of them had had any hypoglycemic unawareness events since their transplant. The safety profile of Tego was presented, which is looking very, very good in that patient population, with really no serious side effects, no serious opportunistic infections. So the data there looked quite good and quite encouraging. I mean, not only has Dr. Wachowski been presenting that data at many, many diabetes conferences in the last 6 to 9 months, but you can actually go on TikTok, and the patients that are in our study are actually describing almost day to day how well they're doing on our drug after their islet cell transplant and commenting that they're able to eat cheeseburgers for the first time in 30 years. So the patient experience thus far has also been quite encouraging.
What are your hopes for patients in the long term?
So I think our goal and our mission, which is actually starting to look like we're making some progress in the last couple of years, is really to replace tacrolimus as the cornerstone medication for the prevention of transplant rejection, regardless of organ type, regardless if it's a cell, regardless what species it might be. So we continue to try to build on that, in addition to continuing to pursue our kidney transplant studies and taking over and expanding the islet cell studies that's been done in Chicago. We're also going to hopefully launch an investigator-initiated study for the prevention of liver transplant rejection this year at a couple of sites, hopefully one in Canada and one in the United States. So that would give us another organ besides kidney and islet cell. In addition to that, one of our collaborators has been working on a really challenging and significant unmet need in pediatrics with kids that are born with congenital heart defects that need a heart right at birth. They often can't qualify for a heart because they can't match them with the right size organ, as you can imagine. The kids are put on artificial systems to maintain their life, but the morbidity is incredibly high in the first 12 months. This investigator, Dr. Cleveland at USC, has been transplanting juvenile xeno hearts from pigs into non-human primates and showing amazing data where he's had animals out past 6 months to a year that have survived with a juvenile cardiac pig transplant. He's hoping to use that as a bridge in the clinic here soon, where he could take a xeno heart and plant it into one of these newborn kids that has congenital heart defects and at least get them bridged out to the point where they're big enough and on the donor list long enough that they can then move to an allotransplanted heart. So that could be an incredible movement in the xeno space in the next year or so.
Are there any other improvements in the field that other companies are working on?
Yeah, the amazing thing about science today is it moves incredibly quickly. So we're seeing lots of changes across multiple different transplant fields that we've talked about today. I think we'll continue to see an evolution in the genetics that are modified in the pigs to make them more humanized. I think we're going to see other cell sources besides cadaveric islets to help with patients with type 1 diabetes, because there's only so many pancreases you can get from deceased donors. We're starting to see technologies where you can take what are called pluripotent stem cells that you can differentiate into any cell in the body and drive those cells to become terminally differentiated beta cells that make insulin. And we're going to see some progress. There's been a lot of preclinical work putting those cells into non-human primates or mouse models to cure models of diabetes. I think you're going to start to see those move towards the clinic here. And again, some of the secret sauce we might need from those cells is to protect them from the immune system. So we're hoping again that tegoprubart will be the cornerstone medication that protects those cells post-transplant.
This isn't an issue that's just confined to one country, it's an issue everywhere. Logistically, are you going to be able to roll this out in as many locations as possible?
Yeah, we are, as I've mentioned, we are collaborating pretty much globally with everybody. I mean, preclinically, we've been working globally, even with many of the leading xenotransplant scientists in China. We're hoping to see those collaborations that have been focusing initially on preclinical experiments and non-human primates move to the clinic this year. But obviously, we've been supplying Tego for those studies for several years in China. So we're hoping that that will continue in the xeno space and continue to build on the data that's being generated here in the US. We're also hoping to be working with a collaborative network doing xenotransplant in Europe. They're moving into humans for the first time as well, initially focusing on kidney transplant, and they've asked to utilize Tego in those studies as well. And then, as I mentioned, in allotransplant, our phase 3 study is going to be a global study. I don't know if we're going to end up all the way into Asian countries for the phase 3, but we're certainly going to cover many countries in Europe, South America, North America, and Australia.
As if you weren't busy enough, are there other things in your pipeline?
So tegoprubart's our only drug, but as you can hear, it's a pipeline drug where it can work in almost any type of transplant indication. So there's other significant unmet needs in transplant, including studies that we haven't done yet called switch studies. So as I've mentioned, tacrolimus ends up having long-term toxicity that affects the organ, and a lot of people can't tolerate TAC. So there's no other options for those people. If they get off their TAC because they're not tolerating it, they risk losing their organ. So one of the things that we could do is switch people over to Tego post-transplant. So if patients are experiencing toxicities with TAC, one of the clinical studies that we could do is to switch them over to Tego and show that we can protect those organs even after a switch from TAC to Tego. The other type of issue in transplant is people that are 5 years out post-transplant, the biggest mechanism by which they lose their kidneys, if it's not TAC toxicity, it's a process called antibody-mediated rejection, where antibodies recognize the donated organ and attack it. There's currently no effective FDA-approved treatments to prevent antibody-mediated rejection. And as I mentioned, tegoprubart will inhibit antibody production by B cells. So we think we have a great opportunity to see if tegoprubart can impact later stage antibody-mediated rejection. And so that's another study that would be on our radar screen at some point in time.
What does the next year, 2 years look like at the company?
We have a really busy couple of years in front of us here. So launching the phase 3 study this year is obviously a top priority for us. It's just a lot of logistical work. It'll be a really big study, a really large study, 100 clinical sites worldwide, working really closely with our vendors to make sure we successfully launch that study and execute it really well. As I mentioned, it's going to be a busy time taking over and launching our phase 1/2 islet cell study in people with type 1 diabetes. We're going to have to launch and manage our manufacturing pipeline to make sure we have enough clinical supply for tegoprubart over that period of time. And then, because of the fact that the drug is starting to look so encouraging across multiple indications, we have inbound calls almost weekly from investigators wanting access to our drug for their clinical research programs. And that could be anything from protecting AAV viruses for gene therapy instead of the way that they do it today, all the way to protecting CAR T therapies. Investigators want to use it for other types of transplanted organs that they're doing. We get a lot of inbounds for other types of organs that we haven't thought about at this point in time. So it's going to be a busy few years for the company, I think, as we launch these programs.
With all of those requests coming in, are you able to accommodate them all?
We accommodate almost everyone that we can as long as we think it's good science and we think it's safe. We want to be very collaborative. I mean, that's been the company's goal since we founded the company.
Hugely important and groundbreaking work going on there. Definitely excellent news. So that's it for this week's podcast. If you do want to get in touch with guest opportunities, or if you want to advertise, please feel free to contact us at jim@deeptechdigest.com. So until next time, wherever in the world you are, I hope you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.